Hello. This is your Unit 6 review. Just going to walk you through what we've done for the last couple weeks on genetics and biotechnology. Some of my favorite things. Okay, here we go. And as always, things with stars are things that you should probably pay a little bit more attention to, but they're take a look at all of the slides. So genetics. This is the study of heredity, and that is when traits, which are characteristics like hair color, eye color, your height, all of those things are passed from parent to offspring. In non-human organisms like plants, that could be something like the shape of the leaf or the color of the petals. So those are your traits. And here we talk about a little bit more traits, a few more traits, some kind of crazy ones. But these are examples of traits: the size of a seed, the seed, the shape of a seed. Do they have striations? Do you remember in our Botanis Curious lab where we looked at the shape and the size of the seeds, as well as the shape and the color of petals to see if things were genetically related? You have dimples, all kinds of things. These are all traits that are coded for in your DNA, and they are passed from parent to offspring. Oh yeah, antibiotics. Bacteria could be sensitive to antibiotics or resistant to antibiotics, and those are also traits. It's not just humans. Plants, bacteria, everything. Anything that has DNA is going to have traits that are coded in their DNA. You can watch these fun. You guys love these little people. Take a moment to watch this if you want a refresher on traits. Variations, these are just differences in traits. So for example, the spotting pattern of these three offspring, well, these are six. There's the mom, offspring 1, offspring 2, offspring 3, offspring 4, offspring 5. They all have slightly different variations of the trait, which is the color of the shell. Alleles, don't spend too much time on this. These are just different forms of the inherited characteristics. For example, if the gene is for eye color, the allele would be the genes that are coding for a particular eye color. Like if you get the blue allele from mom for eye color, you get the brown one from dad. So they're different forms of the same gene. And they're always lined up together on chromosome pairs. But don't worry too much about alleles. I just want you to be familiar with that word, that they're different forms of an inherited characteristic. All right, you don't need to do this. We did that already. This was kind of fun. The ability to taste PTC is a genetic trait. Some of you guys had it and some of you didn't. And that is encoded for a gene that has a receptor. Hey, where did that one go? There we go. Oh, I mixed these two up. All right, anyway, watch these little wooden people if you want to. They're more about traits. I think this was your favorite one because everybody liked the look on this one's face. Okay. So the PTC gene, this is just an example. I don't need you to know any of this information really. All I want you to know is that if you had a particular gene, in this case it was the PTC, you have the trait for tasting PTC. And what happens is that PTC gene encodes a protein. That's what all genes do. They encode proteins. In this case, this particular protein is a receptor that happens to sit right on the surface of your tongue. And if you have the PTC gene, your receptor is shaped like this. And that means the PTC can come onto your tongue, bind to the receptor, and send a signal off to your brain that says, that tastes disgusting. If you do not have the PTC tasting trait, your receptor is shaped differently. Now remember, shape, when it comes to proteins, is the most important thing about how it functions. So if this PTC receptor is shaped a little differently, say there's that little bump in it, now the PTC can't bind, so therefore there's no signal getting sent through this receptor to your brain. So even though the PTC is exactly the same, it doesn't fit this receptor, therefore it doesn't send a signal to your brain. So the PTC trait is one that is inherited and encodes for a protein that helps you taste these bitter tasting compounds. I don't think I have any other thing to say about that. That was just sort of an example of what we're doing. So how does DNA relate to traits? meaning DNA, that molecule that's in every cell of your body in the nucleus. How does it relate to traits like brown hair or blue eyes? Excuse me. The traits are coded for by the sequences in your gene called genes. So these little fragments of DNA that help form these traits, and they do so by making proteins. So if you have the gene, this is just some, I don't know, ways to keep the language straight. So if the person has the gene for PTC testing, there'd be a piece of DNA in all of their cells. They will have the trait to taste PTC, and so on and so on. The genes encode for the traits. All right, so chromosomes. What I need you to know about this is that chromosomes are located in the nucleus. Here's a cell, and in the middle of every cell, at least every eukaryotic cell, there's a nucleus. And in the middle of that nucleus, you have how many pairs of chromosomes? Did you say 46? No, wait, 23 pairs, 46 chromosomes. So you have 46 chromosomes in every nucleus. And in every nucleus, and every chromosome is made up of DNA. So the DNA is wound around the chromosome. And if you were to unwind it completely, you would see that it is made with all of these base pairs, A, C, G, T, all along. It's in a double helix form. I'm going to talk about that more in a second. But so little parts of this DNA, for example, I have this little funny looking bracket I tried to make, but little parts along this DNA are genes. Maybe this part of the DNA codes for your eye color. And maybe one down here codes for your, I don't know, what else do you have? Your ability to taste PTC. And maybe one down here codes for the structure of your neurons. All of those genes are wrapped around super, super, super tight around these histone proteins. They form these chromosomes, and those are all tucked neatly inside the nucleus where they stay. Those are the instructions to carry the hereditary information those are the blueprints for your body, your specific body. Nobody else on the planet has your same DNA. So genes carry the information, and they're found at specific locations on the chromosome. You might see a chromosome depicted like this. Sometimes you're going to see it looking like this. Sometimes you're going to see it looking like this. And you can actually show where on the chromosome those traits lie. And take a moment to watch this video if you feel like it. All right, DNA, deoxyribonucleic acid. This is the genetic information. It encodes all of your traits. You should all know that by now. It's made up of 4 bases, A, C, G, T. You don't have to know their chemical names, although you can if you want. That could make a good bonus. Just saying. All right, so it has a double helix structure. Don't waste any time. Don't memorize these. I'm just kidding. Don't waste any time memorizing those. It's not going to be a bonus. All right, so it has a double helical structure, and along the middle you have ladders of A's and T's paired with C's and G's. So you're never going to get, here we have this code, adenine and thymine paired together. A's and T's always go together. C's and G's always go together. Okay, and then there's this phosphate backbone. DNA must be replicated before every single cell division. And this is just an image I wanted to show you guys about that. So imagine this is the DNA before it's copied. It's a double helix with C's and G's paired up and A's and T's paired up. Then while it's replicating, it unwinds and makes a new strand. So once this is done copying, you're going to have one new strand here. Where are we? And then another new strand here. So this is just a picture of what you might expect to see if you were getting a question about what DNA replication looks like. Again, this happens every time a cell is about to divide, because if the cell divides and each daughter cell gets half of the DNA, you're going to eventually run out of DNA. So it has to replicate itself every single time before it divides. This is DNA replication. All right, and the whole point of this anyway, all of your genes, all of your DNA, they're the instructions to make proteins. That's the point of every gene you have. It just makes a protein. Every gene encodes for a protein, because proteins are receptors, enzymes, antibodies, signaling molecules. They essentially do everything in your entire body except for like a few things. They're structural. That means they give you form. They're functional. That means they have very important jobs. They help you digest your food. They make your energy, help your cells divide. So that's why we're doing this. All those genes are they're just code for proteins. And this is just kind of a little, well, I'm not even going to talk about that, but just a little side note there about where we get our protein. You can watch this video if you want. This is a good, oh wait, I didn't mean to start it. Okay. This is a good overview of the basic function of DNA. And here we see the comparing and the contrasting of DNA and RNA. I'm going to talk about RNA more in a second. But so here's DNA, or double helical structure with A's, C's, G's, and T's. RNA is the second step in this process of making proteins from our DNA. It's sometimes called mRNA or messenger RNA. But instead of A's, G's, C's, and T's, we have A's, G's, C's, and U's. Okay, so we have uracil instead of thymine. The two differences, just some differences. DNA is the same in all cells of your organism. And RNA is only turned on when the specific protein is needed. You don't need to know the kind of sugar, but that's just the sugars that make up the backbone of the molecule. Sorry, can you hear my dog? Okay, so A's, T's, G's, and C's are DNA. A's, U's, G's, and C's are RNA. DNA is double-stranded. RNA is single-stranded. There's only one kind of DNA in your whole body, but there are three kinds of RNA. There's mRNA, which is the one we're really talking about. We'll also talk about tRNAs a little bit. They help add the amino acids to the protein, and ribosomal RNAs are part of the ribosomes. Okay, so the steps in making a protein. First, you're going to make the RNA, the mRNA, and this is by a process called transcription. The mRNA is a single-stranded copy of the DNA that can leave the nucleus. So the DNA is always in the nucleus, but the mRNA can get exported out of the nucleus, where it gets turned into a protein. So then the mRNA is translated by the ribosome. I can't even stop this. All right, so just ignore my dog. All right, so the mRNA is then translated by the ribosome to make a string of amino acids called a protein. And then this protein is folded into a specific shape. I'm going to say this probably 17,000 times because the shape is what determines the function of the protein. So you first have DNA. First you're going to start with DNA. Here we go. Double-stranded. That gets transcribed into mRNA. We know there's going to be U's in here instead of T's. You're never going to see a T in an RNA sequence. And then using the genetic code, that gets translated to the protein sequence that is made up of a chain of amino acids. Amino acids are the building blocks of proteins. Okay. So translation. When the mRNA is translated into a protein by ribosomes, the ribosomes read the RNA 3 bases at a time. So it doesn't read the whole RNA like CG, UA, G, blah, 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 blah. It reads it in 3s, CGU, GAC, AGU. So it reads it in these triplets or these codons, and each codon has a specific amino acid that it's matched to. These amino acids get added together into a long chain. They form a protein that then what? What happens to it after you have a long protein? It folds into a specific shape where it functions and then contributes to the trait. Here's the genetic code. When you finally have your RNA sequence, you're going to find your RNA codon on here. You can use these first, second, third letter guides, or you can just scan here to find the appropriate amino acid. But then you can figure out the protein sequence. I'm going to have you guys do a quick test here. So pause it in between. I'm just going to keep talking because I apparently need to take my dog out. So just pause it in between. Here's the DNA sequence. Pause it and write the RNA sequence. Okay, and then when you unpause it, did you unpause it? This is what the RNA sequence should look like. Now you're going to pause it again in a second and write the amino acid sequence. And this is what your amino acid sequence should look like. It always is going to start with a methionine. This is the start codon, AUG. It's always going to start with a methionine, and every protein always ends with a stop codon. So there could be 3 different stop codons. You don't need to memorize any of the stop codons, but you might want to know AUG because that could be a bonus, right, Lindsay? All right. Just more fun stuff if you want to talk about if you want to learn more about transcription and translation. These are some of those interactive things that we did. All right. If you want to know if organisms are genetically related, you can use gel electrophoresis, also known as DNA fingerprinting. This is like, remember we cut those fragments in for our Botanis Curis lab? And we've done a couple of these problems in our do nows or homeworks. Essentially, you're going to run a gel electrophoresis is where you apply electrical current to this gel. Positive up here, negative up here, positive down here. The DNA is negatively charged, so it runs toward the positive end. And the smallest fragments are going to be down to the end. So if I asked you which one of these fragments is the smallest out of all of these, you'd say, well, it's that one because it ran the farthest. And if I asked you which one was the largest out of all these, you'd say, well, it's that one because it didn't run as far. So we used restriction enzymes. Remember, we pretended our scissors were cutting between the C's and the G's, double C, double G. Those were supposed to mimic a restriction enzyme that cuts the DNA at specific parts, specific sequences. And then you compare the fragments. So you would look to the mother, father, and child, and whichever has the more similar fragments are related. So you can see that the child shares fragments with the mother and the father. Okay? But if we were comparing these three, sometimes I might give you an example like our Botanis Curis lab where there were two samples that were much more similar than the other in their DNA fragmentation pattern. So those would be more likely to be genetically related. That's how you can tell. All right, mutations. Mutations are changes in the DNA sequence. Sometimes they happen if when the cell is replicating its DNA every time in between cell divisions. Sometimes the DNA makes mistakes. The DNA replication makes mistakes and they're not caught by your repair machinery. So that's where you can get a mutation. They usually get repaired, but sometimes they don't. And most of them don't actually make a big difference because there's a lot of your DNA that doesn't really code for proteins. They do other things. So DNA, you can get mutations two ways. Your DNA fails to copy accurately, or environmental. The environment can also create mutations. So, for example, mutations can be caused by exposure to chemicals, X-rays, ultraviolet light, radiation. All of these things cause the DNA to break down and cause mutations. And there are three types of mutations. One is a substitution, where, for example, let's look at this as our original DNA sequence. A substitution would just be when one base pair was replaced by another base pair. See, you have a G, and it's going to a C. Oh, my dog just walked away. I think he's mad at me. Sorry, Bear. Okay, so, and then there's an insertion. An insertion is when you have an addition of a base. A base is added in. So let's see, here we have 1, 2, 3, 4, 5, 6, 7, 8, 9 base pairs. And here we have 1, 2, 3, 4, 5, 10. So that's how you can tell if there's been an insertion. You can really just count the base pairs. You don't have to really look at the exact sequence. And the same goes for a deletion. If you have fewer base pairs than you started with, that's going to mean you deleted a base or deleted a nucleotide. So those are the three types of mutations that you can get. So they can be caused by failure to copy accurately, just mistakes during DNA replication, or the environment. I feel like I'm missing something on that one. All right, how do mutations affect the protein? Well, the DNA mutations change the amino acid sequence. You're going to change one of these base pairs, you're going to have a different amino acid that's going to be added. And you can play around with this interactive if you want to. It shows you exactly. You can make very specific changes to the DNA sequence by clicking on Edit DNA. And then you can click on Show Protein, and you can show how very different one base pair change can be, especially if that base pair change is in a stop codon or creates a stop codon or creates a frame shift, which is where you're completely changing the triplet code that you are reading. So you're changing the pattern of triplets if you add or subtract a base. You don't need to know about that too much, but we did talk about it. I just think it's kind of interesting. So anyway, when you change the amino acid sequence of a protein, you're going to completely change the shape, which completely changes its function. All right, mutations can only be passed to offspring if they occur in your gametes. If you get a mutation in your skin cells or your eye cells or your intestinal cells, they cannot be passed to your offspring, but they can be passed to the cells that they divide and become. So when they divide by mitosis, which all of the cells in your body generally do, the daughter cells of that one cell are going to have that mutation, but it's not going to go to your babies unless it gets into your gametes. These is just some kind of fun facts about how to prevent mutations. And gosh, I hope I'm not going on too long for you guys. But anyway, bear with me. So the environment can affect when genes are turned on and turned off. So we know the DNA is the same in every cell of your organism, and they're turned on and off as needed by the cell. This is called gene expression. We talked about this last unit. So what makes them turn on and off? Well, there are a bunch of different things that can in the environment that can influence gene expression. Genes can be turned on and off by things like light, hormones, vitamins, exercise, drugs, chemicals, also the position in the embryo, where the cell is in the embryo. Remember when cells are like in a ball? Depending on what side of that little ball you're on, actually could affect what cell type they become, what genes get turned on and off. Anyway, just a couple vocab words before we talk more about that. Genotype is the DNA, the genes you have. Phenotype is the way that they look. So it would be you might have the gene for, let's talk about the Arctic fox. Maybe he has a gene for black fur, but in cold temperatures or in low sunlight, he looks white. So his genotype would be one thing, but once you influence it with the environment, you may produce a different phenotype. The phenotype is really the traits that are shown or how things look or how they're exhibited. Okay, so here's an example. I'm not going to go through these too much because I feel like you guys, this is pretty straightforward. I'm just going to choose one example. So here's an example of light affecting the genes that are turned on by this plant. Here's another example of temperature affecting the genes are turned on by this bunny. This is, I think, a great example. This bunny, I think the scientific word for him is Himalayan rabbit. This Himalayan rabbit, over 30 degrees is all white. But if this guy is reared, that means raised at 20 degrees or less, he gets black ears and a black nose and black feet. And that is because you're turning on the gene, the cold temperature helps turn on this gene for black pigment. And that, as you guys all guessed, helps him absorb heat in the places where he might be the coldest. So temperature can play a role in expression of genes. All right, see if you remember this experiment. This just proved that temperature was playing a role in the expression of that pigment gene. We have the Arctic fox, same kind of thing. Different. This was about times of darkness, times of light. When it's too dark, this guy, I'm not going to go through all these because but anyway, you can read through these. These are pretty self-explanatory. And you guys also kind of had a good idea about how that would work. pH can affect the expression of pigment genes in flowers, like these beautiful hydrangeas. pH is how acidic things are. If it's a low pH, 4.5, that's very acidic. You're going to get a different color than you would get if you brought the pH up to 7, which is a neutral pH. That would be pink. Alrighty, twins, they have the exact same DNA, and they are not exactly the same, even if they grow up miles and miles apart. They can be similar, but they're not going to be exactly the same. That's because their environment also affects their traits. Okay, now we're going to move on. I think this is the last part of the unit. There are 3 processes of genetic change or modification, and we're going to go from the oldest. In fact, this is so old, it was before there were even a thought of humans on the planet. Selective, but although it still occurs. Selective breeding began thousands of years ago, maybe 1,000 years ago, about 1,000 years ago, maybe. And genetic engineering, this is the most recent, this is the most modern technique for changing the genes of particular organisms. So you can read through those types, but I'm going to discuss each one of them individually. Natural selection is the process by which, according to Darwin's theory, only the organisms best suited for their environment tend to survive. And then they transmit their genetic information, that's their DNA, in increasing numbers to succeeding generations, while those less adapted tend to be eliminated. That means they die. They die out or they become extinct. So what's a great example of this? The peppered moth. So as we discussed, the peppered moth comes in two colors. So it's two different traits. One is for like a white speckled color, and one is for a black color. And there was a time when most of the trees in this area were a light colored tree. So if you're a bird, which moth are you going to go after first? Well, you're going to probably see this guy faster, right? So there were more speckled white moths because they were able to blend in with the white trees, avoiding predation, that means getting eaten, by birds. Therefore, they could survive and pass along their traits for speckled white wings to their offspring. But after the Industrial Revolution, the burning of large amounts of fossil fuels left black soot on all the trees. So after many months, there was a shift in the population of peppered moths from the speckled guys to the black ones. And eventually the black ones took over the majority of the population. And this is because now they were able to camouflage with the trees, avoid predation, and pass on their genes for black wings to their offspring. So they kind of overcame the lighter colored ones as the predominant trait for that species. So here's a couple more pictures of those. I recommend you go to this. If you're interested, you can go to this website, read more about them. Here they are in the light-colored tree. Here they are in the dark-colored tree. And poor this little guy is going to get eaten now because he doesn't really camouflage anymore. So natural selection is just about traits that are just naturally beneficial to allow an organism to survive, just being passed down to offspring in greater numbers than traits where the organisms are getting eliminated more frequently. Here's another example. We talked about that before. I feel bad for that little guy. Anyway. And take a look at this video. Natural selection. These dark colored frogs probably blend into the log a little bit better, so they tend to survive more. And these little guys might get eaten by this bird. But you can watch that video. Okay, so number 2. So natural selection has been going on since the dawn of life. And number 2 is selective breeding. This has been going on for thousands of years. And this is first when humans started just really domesticating animals. And it's a process by which humans use animal breeding and plant breeding to selectively develop particular phenotypic traits by choosing which typically animal. Oh, see, I meant to edit this and I didn't. By choosing which animal or plant males and females will sexually reproduce and have offspring together. So basically, you're choosing the parent cells or parent plants or parent animals that have the traits that you want to see in the offspring, and you're choosing them specifically and having them mate together. There's my lovely little example, but it's essentially you're just looking at examples like this. Horse breeders, they would choose the 2 fastest horses to breed to the offspring. So the offspring will also have the trait of running fast. If you were a horse breeder, would you choose 2 kind of slow old horses and expect to get a fast horse? No way. You choose the traits that you know are most likely to be passed on to create the phenotype that you want. Dog breeders will choose male and females to mate based on their coat colors to produce offspring with the desired traits that they want. That's a big business. Plant breeders will choose 2 plants that seem to be resistant to pests and cross them and see if they can get offspring of plants that are resistant to pests. This is a very kind of simple but obvious technique. You're just going to choose, well, it wasn't obvious when they didn't realize that DNA was like hereditary, but You're going to choose animals with the traits that you want, breed them specifically, and then they're going to pass that down to their offspring. All right, last but not least, one of my favorite things, genetic engineering. And this involves genetically modified organisms and cloning. So today we talked about genetic engineering of genetically modified organisms, particularly fruits and vegetables and food. But genetically modified organisms are any organisms or plants or microorganisms in which the genetic material, just the DNA, has been changed in a way that does not occur naturally. So selective breeding and natural selection, those just both kind of occur fairly naturally. Selective breeding has a little bit of a push, but when you're talking about genetic engineering, we're talking about manipulating the DNA. Humans are manipulating the DNA of other organisms in ways that would never occur naturally. This is just a general overview. You don't need to know all of this stuff, but basically what you do is you identify a trait you're interested in. Let's think of one. Let's say, what did we do? Let's talk about resistance to being eaten by pests, because that was one that we kind of talked about a little bit today with the corn. We would identify that trait, so we would take that maybe out of a bacteria or some other cell. We found another type of cell that was resistant to pests. So we take the gene out of that one, in this case a bacteria. We'd isolate it using enzymes, and then we would insert it into the new genome. So say this was the corn genome. Corn genome is not a circle, but we're just going to pretend. And we're going to take the pest-resistant gene, and using enzymes again, we're going to put that piece of the gene into the corn genome and then insert that genome into a new seed or a new cell and have it grow up with the new trait. So genetic engineering is essentially taking genes out of one organism and putting it into another one. We're going to probably watch this tomorrow. This is a cute little rap these kids made up about plasmids. Plasmids are these circular pieces of DNA, so-called cut bacterial DNA, they call that here. And here, it's kind of hard to see, but there's a little tiny piece of DNA. That's your gene of interest. Say, in this case, it's the bacterial resistance, the pest resistance gene. Sometimes you'll take that gene and using enzymes, you'll put it in the plasmid DNA to get this circular piece of DNA, which then you insert into a bacterial cell, and then you kind of hijack the bacteria and you kind of trick the bacteria into making that pest resistance protein for you. This is done for human health purposes too. So for example, genetic engineering, recombinant DNA, these are sometimes they call, recombinant DNA is kind of like saying genetic engineering. It's kind of like when you're making plasmids and doing all this stuff, they sometimes refer to it as recombinant DNA. So I just want you to be familiar with that term. That's kind of like when you're mixing different pieces of DNA together. So genetic engineering is used to produce the whole human hormone insulin. And this is a classic example where the insulin gene is taken from a cell of a human, could be anybody, and it is cut and pasted using enzymes to insert it in a round fragment of DNA called a plasmid. It's just like this. You may see it like this. So now we took the human genome, we clipped out the insulin gene using enzymes, and we're going to plop it into this plasmid. Then we're going to take that plasmid and insert it into the bacteria. And they're going to replicate like they're just replicating on their own like they would normally do. They don't really even know that you kind of snuck this human insulin gene in there. It's very, very sneaky. But you got them to produce human insulin, the human insulin protein, without even really knowing it. And then you extract the human insulin protein. This is actually the human insulin protein. These are all the individual amino acids. It's 2 chains and they're held together by these disulfide bonds. Anyway, so you've tricked the bacteria into making human insulin, and then you can extract it and inject it into a person who has problems with blood sugar. So that's a great story. And this used to come from pigs for diabetic patients. What's one advantage? Why wouldn't you want to necessarily use a pig protein in a human? Think about that for a little bit. All right, and this is something else we're going to talk about, but I just want to go over it for this video. Cloning animals, making exact genetic copies of an animal, of an adult animal. So they have the same DNA. You know that you don't have the same DNA as your mom or dad because through the process of sexual reproduction, you get half the DNA from your mom, half the DNA from your dad, and you get new combinations of genes that have never been seen before. But in this case, you're actually completely cloning. You're going to make a genetically identical organism. So you're going to take, sorry, I'm getting distracted. My dog is barking again. Okay, so you have this female sheep. You're going to take a body cell from her. Say, let's take it from her, I don't know, maybe from her intestine. I don't know why. You're going to take a body cell from her. It doesn't have to be a gamete. In fact, it shouldn't be a gamete because her gametes are only going to have half of her chromosomes. She needs her full set of chromosomes if she's going to be genetically replicated as a clone. So you just take a random cell from her wherever. Let's take it maybe from her mouth. Remember when we took cheek cells from our mouths? Let's pretend we'll do that from her. We'll just take a random cell from her mouth and put it in a dish. And then you're going to extract the DNA from that. You're actually going to do this using a tiny, tiny needle that sticks right in there and sucks up this, and you're going to do it under a microscope. The needle is smaller than you could see with your eye. You're going to pull out this DNA, right? And you're going to keep it in your needle and don't move. And then on the other hand, you're going to have an egg cell that you've extracted from the ovary of a completely different female. And you're going to take out the DNA out of that one. And you're going to just so you're going to take the DNA, and that DNA you're just going to throw in the trash because you don't need it. And you're going to have this empty egg cell. Now what you're going to do is you're going to take the full set of chromosomes from this female and put it into the empty egg of this female. So now you have a full set of chromosomes in an egg cell. You kind of just mimicked fertilization, because in fertilization, where you have half the set from the mom and half the set from the dad, you're just faking it. You're taking the whole set from the mom and sticking it in the egg cell. And then you're using hormones and things to try to get it to start replicating and pretend it's an embryo. And it actually works. So when it's about 100 cells or so in a dish, maybe not that many, I don't know. But when you have this is called the blastocyst stage, you're going to take that embryo, you're going to implant it in another sheep's, are these sheep? Yes, another sheep's uterus, where it's going to implant in the lining of the uterus and continue to grow until it becomes a fully developed lamb, which will be born. But it had only the DNA from this one organism. So they're going to be genetically identical. And this does happen. The first time it happened was in 2007. Dolly the sheep, she's famous. But all of these animals you see on this page have all been genetically cloned successfully since 2007. Not a lot of them, but they have. So humans are not really cloned. And what you should know about that is there's a big ethical issue with cloning humans. Some people feel it's not right. South Korea did claim to have cloned humans quite, like 10 years ago, but that was proven to be false. We're going to do this tomorrow. You're going to pretend you're cloning things. But I think that's it. Let me know if you have any questions. You can send me a message on Remind or talk to me in school tomorrow or whenever. All right, take care. Have a good night. Bye.